You're right.
Hello, people in the back. Can you hear me?
Let me just.
It's echoing.
Let's try again.
Okay, we're trying again. Can you hear me Can you hear me now? Apparently not. Can you hear me? Like clearly or a little soft or? Okay.
We don't have to yell.
Oh,
Here we can maybe just bring this up. So if you're here, it's going to be all better. Just go outside to check on my food. Do you need a pointer?
probably.
People sometimes use the computer.
Okay.
Yeah, we will see it here, but the Zoom people will not see this one.
Oh, yes. Yeah.
So
yeah.
sometimes people use on the PC, if you right click, you can choose pointer.
Is that anything useful? Pointer options,
Yes, okay.
automatic hidden
So
visible.
that means
I
I
don't know.
have.
Visible? There's a pen thing. So I don't see the doodly doodly. But that's okay. This
Are you
thing
gonna
will
be
work.
okay?
I think so.
Okay. Let me just go outside and check.
Okay. Oh, now I'm in trouble. We're roles are reversed here. Now, yeah, right. I got you. Okay. I'm quaking in my boots. Are you going to heckle me?
I'll turn this off now.
Oh, but
Okay, good afternoon, everybody. Thank you very much for attending. It's my great pleasure to welcome Dr. Marra to the CBR. So Dr. Marra is a full professor in the Department of Medical Genetics and a member of Michael Smith Laboratories. Marco completed his PhD at Simon Fraser University, followed by a postdoctoral fellowship at Washington University in St. Louis, Missouri. Dr. Marra is a key driver in genome sciences in Canada, and his list of accomplishments is far too lengthy for me to go into in detail. But a couple of highlights is that Dr. Marra was inducted into the Canadian Medical Hall of Fame and has been made an officer of the Order of Canada. So we're very, very honored to have Marco speak to us today. And Marco, welcome and thank you very much.
Well, thank you very much, Hugh, for those words of introduction and for offering the opportunity to tell you a little bit about an area that I'm passionate about, and that's precision cancer medicine. And so today's story is going to be a bit of a historical recount, a little light on data and a little heavy on a tale. And the tale is one of a grassroots initiative that started 15 years ago with an early observation, and I'll describe that, and that has grown into a national program that has been recently renewed by the government of Canada, despite all the financial hardships. And so it's been a struggle, and there are encouraging signals that go along the way, and we worry about the continuation of what we're doing and its impact on patients, hopefully positive and hopefully not too long from now. But the realities of our situation are that we must persist, and indeed in all of our research activities that we all engage in, we must persist. So with that, we'll talk about POG, BC's Personalized Oncogenomics Program, and how it helped drive and now participate in the Terry Fox Research Institute, Marathon of Hope, Cancer Centers Network. That's quite a mouthful, but there you go. So I do a lot of things with TFRI, and I get money from them. And so I offer this disclosure so that you can evaluate my words against all these things that I'm involved in. So I thought it would be useful to start the presentation today with a quote. And I like this quote. It's by Siddhartha Mukherjee, who is the author of The Emperor of All Maladies. And I don't know if anyone has had the opportunity to read this book, but I'm seeing some nods. Thank you very much. It's an interesting treatment of cancer, and the new edition has just come out, apparently with updates and new chapters. If you are interested in the area and its impact on people and a well-written book, you might be interested in this one. So the essence of the quote is that, an individual's cancer is a unique thing. And we all can fundamentally understand that when we realize that the pattern of variation in our normal genomes is extensive. We haven't even written it all down yet, but we know that a cancer that arises from within us carries footprints of that genetic variation that we all carry in our normal cells, as well as some other changes in the genome that arise as a consequence of cancer properties that I'll describe. So this business of the individual nature of cancer patients and their cancer is really what has driven a lot of the questions around cancer most recently. And I would say, reflecting back on 25 years of cancer genomics, or thereabouts, I think it's fair to say that the single biggest contribution that genomics has made in our understanding of cancer is the incredible heterogeneity that cancers represent. And this challenges us to think about ways we can treat a fundamentally heterogeneous condition, imagining back, if you will, to that point in time when we imagined that there would be a magic bullet, something that we would give cancer patients and it would go away. I think that's still a formidable challenge and maybe not feasible. Anyway, on the business of heterogeneity, I offer these two pictures The references are given there if you want to read more about them. So the circle thing is a depiction of cancer. And this has been published in 2 papers about a decade apart by Hanahan and Weinberg. And so at the center of this is a cartoon which depicts a cancer as a collection of diverse cell types and tissue structures. So we've got some vessels there, we've got some stromal cells, and we have some tumor cells. And so this is not a homogeneous entity. It is an admixture of features of cells and of tissues that together operate as part of the tumor mass. So it's a heterogeneous mass of stuff. Surrounding this central picture is a list of properties, if you will. And I don't even know if they're readable. I guess they're more or less readable if you've got good eyes. Matthew, can you read those things? Okay, I can't see you, Matthew. So I'm assuming you're nodding. Yes, you can. And right down here is genome instability and mutation. So there's all these properties, including genome instability and mutation. And then around the outer edge are a heterogeneous collection of classes of compounds that can target vulnerabilities that are thought to emerge from cancers. So a heterogeneous tissue mass surrounded by a heterogeneity of properties that drive malignancy, and then a heterogeneity of treatment options that you might consider. It's all about difference. On the right is a depiction of some of the ways cancers can be different. And so we see in A, spatial heterogeneity, this particular example, the concept of multiple metastatic lesions within an individual, and they differ in the composition, in this case, of their cells. So we see this within a patient. And then if you imagine across a patient trajectory, and that's what the temporal heterogeneity picture is meant to illustrate, we can see cancers changing over time. And so this heterogeneity that we see in cancer is not a static entity. It is a dynamic property of cancer. And if you want to think about it in terms of evolution, think about it in terms of evolution on steroids. Within the lifespan of a particular cancer patient that undergoes serial treatments, there will be selective pressures placed on the cancer cells in the tumor, and cancer cells that are resistant to those selective pressures can expand and fill a tumor, a new tumor niche. But in so doing, you create a cancer that is resistant by definition to the prior therapy. All right, so it's not just the properties of the cancer cells that change, and we can read out some of those properties in terms of their genomes or their methylomes or their transcriptomes, what have you. But it's also the composition of the so-called microenvironment, cells that are in the vicinity of the tumor. And you can think of the immune compartment as being amongst the microenvironment cells. They can change and do change across different treatments alongside the cancer cells. So we have this hypervariable dynamic condition. And when we think about therapy mapped on top of this thing, the question is, how do we think about that? And we're not there yet. And I'm not going to solve the problem for you in this seminar, but I want to lay out the challenge, because we are so rudimentary in our beginning to probe heterogeneity in cancers. I think there's a long way to go and ample opportunity for contributions to be made here. So this all seems fine. The composition of the tumor changes, the composition of the microenvironment changes. And we then have to think about, well, what is the capacity of a tumor to give rise to new variants? Remembering that genome instability and mutation is a driving property of cancer. Well, if you just think about the mass of cells that can be in a tumor, you can get about 100 million packed cells in a cubic centimeter, about the size of a sugar cube. And if each of those cells is mutationally unstable in some way or chromosomally unstable in some way, the opportunity for large numbers of changes to occur just as a consequence of cell division is there. And so you can emit every cell division, cancer cells that are different from the parent cell that offer the opportunity for a new path for treatment resistance to arise. It's a formidable challenge. Now, the POG story that I'm about to tell and the Marathon of Hope do not delve into this at the level of the single cell. They're at a rather more rudimentary level, but there's plenty of opportunity to see heterogeneity even when we take a mass and grind it all up and sequence its DNA. Now, what we can imagine emerges from such an exercise is a measurement of averages, if you like. So dominant cell populations, dominant mutations, dominant changes in that blended sample now emerge as evident. So what heterogeneity do we see there? And I'll offer an example here. This is in the context of B-cell lymphomas. This thing is called an oncoprint. Have people seen an oncoprint before? Raise your hands, please. A few people have, okay, many have not. So this is in the context of follicular lymphomas, diffuse large B-cell lymphomas generally. And along this axis on the bottom here, we'll see some yellow and some blue and pink ticks. Those ticks indicate different cancers. And so the yellow is follicular lymphoma. We have some frequently mutated genes on the y-axis. And every time a patient showed a mutation in one of these genes, we make a little tick. So we see the ticks going up. We see mostly blue and some green and a few red. And those ticks just mean different classes of mutation are found. And the way to read this, starting from the patient at the bottom, the first yellow tick, read up, and you'll see a pattern of mutation associated with this particular patient. And if you go to the follicular lymphoma right next to it and you read up, you'll see a subtly different pattern. They're not identical at the level of even a few genes that we've cherry-picked for purposes of this example. So that is heterogeneity. And then across the spectrum of B-cell lymphomas, if you look right at the very top, you'll see MLL2. We don't call it that anymore. We call it KMT2D. And KMT2D appears to be frequently mutated actually across a range of different lymphoma types, the yellow guys and the pink guys. So there are mutations that cut across cancer subtypes within the B-cell lymphomas. And then there are unique things that we see only infrequently or sometimes only once in any given cohort. And this pattern is repeated in virtually every cancer. You get a few highly mutated genes and you get a long tail of a distribution with relatively few, but still important genes that emerge. So the clinic, for the longest time, has recognized that there was mutations, and increasingly with the development of targeted therapies, the idea has been we ought to be able to design a therapy that targets a frequently mutated entity in a genome. And so alongside this realization in the emergence of targeted agents against largely oncogenic alterations, there's been these things called panels that have been created. And so a panel is somebody's best guess as to some number of genes and or regions within those genes that ought to be interrogated as a necessary precursor to prescribing a therapy. And so these panels, some of them are shown here. These are ones that are used at BC Cancer. We see something called an oncopanel, and there's some genes and some variants on the oncopanel. We see a focus panel, slightly different. And then we see a hereditary cancer panel, which has a whole slew of stuff on it. And this is the relatively current depiction of the state of things. And if you want to know more, you can go to the cancergenetics.ca website at BC Cancer. And what you will find, if you pay attention to this website long enough, is that these things change. They're not static, because the more we learn about a malignancy, the more we understand that we ought to be looking at this gene or this mutation or that mutation, if we are to align a patient to the best therapy for them. So this business of serial identification and qualification of panel members creates a complication, and it becomes serial. And it becomes very expensive, because every time you have a new addition to a panel, you have to think about how you're going to validate it in a clinical context. And that's not a cheap exercise, and it's a non-trivial exercise. It also raises the potential that these are based on some of the most important mutations that align to therapies. Not all of the mutations that align to therapies. We don't know what those are. And many of these kinds of assays are restricted in their application to certain cancer types. So a panel may be run on a lung cancer case and not run on a pancreas cancer case, simply because the indication isn't there. And this really was the birthplace of thinking about whole genome analysis. And so what if we're not smart enough to guess what all the genes ought to be on any given panel. What if we just adopt a brute force approach and sequence them all? And so we had an opportunity thrust upon us around about 2008 to do exactly this. And this is, 2008. The technology is not what it is today. We did not do this, and we weren't entirely happy about accepting the assignment, but we did so nonetheless. And so what we found is shown on the left panel there. So this is a pretend pathway diagram, greatly simplified. And so there was a case, a rare adenocarcinoma at the base of the tongue with lung mets. And the patient, being a very sophisticated individual, caused us to have a challenge put in front of us, which is figure it out and tell us what the therapy ought to be. And so there were genome and transcriptome sequencing experiments done on the primary, the adenocarcinoma at the base of the tongue, and from the metastatic lesion in the lung. And not only genomes, but transcriptomes were done as a way to gauge whether or not activation of pathways was occurring. And what the evidence pointed to was a gene called RET, which is shown high in the cascade in the is that purple? Purple box? It says RET there, not PTEN, but RET. And the signals pointed to RET. It said RET was up. Well, for this particular patient's cancer, there was nothing that targeted RET. But at the time, in clinical trials for kidney cancer, there was a RET inhibitor available. And through various manipulations that I'm not aware of, other than the result was the patient got the RET inhibitor and applied it, and there was both a clinical and radiologic benefit, and it was extended for some period of time. This was unbelievably lucky. And so it's luck that showed us that such a thing was possible, at least in an anecdotal example. So that was eventually published with great wrangling of editors across multiple journals, felt that this thing wasn't really even ethical, despite what the REB had to say. There was a lot of nerves about this, actually, and we felt it was important to get it out, and eventually we were able to do that. This then, in 2012, around 4 years later, led to the idea that maybe the technology had evolved to the point where maybe we could do this somewhat systematically. And instead of just doing one, maybe we should try and do a few and ease our way into it. But this should be done with clinical rigor, in the sense that these were not so much casual things, but rather had to be endorsed by the REB, as was the first case, needed to have prospective patient enrollment and a plan to do that, and needed to be supported by a consortium of clinicians who could get together and agree that whatever transpired from the analysis was something that a committee of peers had agreed was sound, at least in its rationale. And the population that we were approved to work in were patients that had run the gamut of therapies, had nothing left that could be offered, and many of them were palliative. Very difficult population to work in, and almost impossible to show a survival benefit. But that was the population that we worked in and continue to work in, although there's been some relieving things. So here's the leadership team of POG, and the conspirer, my co-conspirator, Janessa, gets all the credit for everything on the clinical side. It's been a huge lift for Janessa. And she's brought on a team of people from the clinical side that are now contributing unique and novel ideas and side projects to the overall POG program. They're listed here. We have pathology represented. We have bioinformatics represented. We have health economics represented. And the health economics piece, lest I forget to mention it later, is very important, because there is a vision that some version of this will become mainstream for at least some cancers within the span of the next 5 to 10 years. But we have to create an economic argument if provinces are to buy in. So Dean Regeer's work in this regard becomes very, very important. So these are the leaders, and we extended to pediatrics as well. But the team is large, and we're not going to get into it, but it's a large team that spans medical oncology and various other disciplines of BC Cancer. And in fact, the medical oncology component is the largest single component in the mix, and people come and go. And I'll have an enumeration of this later. So what does POG do? we have this circle that we like to refer to when we describe what POG does. And so we have medical oncologist consenting a patient in their clinic. And biopsies historically have been arranged. And this has been a rate limiting step. So biopsies are preferred. These are fresh biopsies taken from the patient. And the reason they're preferred is because they yield the best data. The problem is the biopsy procedures have a risk of morbidity and mortality. They are not casual things. It's a needle core biopsy, often of the liver. And so this is not something that you can just do in a casual setting. There's a risk of hemorrhage and worse. So it's not easy on the patient. So that's not something that we could implement at scale in a population, but it was something that was implemented early on in the program. Then we do this whole genome and transcriptome analysis, the WGTA part, and fundamental to the effort is a data sharing component. So these data are available on the European Genome and Phenome Archive, EGA, and then more recently on the Canadian version called CGA. And so people can request that through BC Cancer. We created a molecular tumor board to review the results, and the results that they reviewed were encased in a report, 2 report formats actually that we created, and I'll touch on that just in a second. Now we're sequencing tumor and normal. And so to the extent the technology allows, we're capturing essentially all, not all, but essentially all of the more obvious variants in the normal DNA. And in those variants are predisposing cancer alleles, moderate to high penetrance cancer alleles. And so we can report those out to the hereditary cancer program at BC Cancer. And the consequence of that is that about 10 to 15% of the cancer cases that we see in POG identify a never before seen familial cancer, so a cancer family. And so you can offer reflex carrier testing to other members of the family, and in this way identify family members at risk. This, I think, is very important low-hanging fruit, and it offers the opportunity for a surveillance that when it finds evidence of an emergent malignancy can think about targeting it safely and effectively to achieve cure early on in the disease course. And this leads to occasionally clinical action. It leads to all sorts of other research in health economics and so on and so forth. So that's basically what POG does. On the issue of the reports, there are 2 that we generate. Now we're reducing essentially 100-fold coverage of the tumor genome, 40-fold coverage of the normal genome, and about 200 million RNA-seq reads down to these reports. And the objective is to do this in clinically useful time. We have a great big long report, which takes PhD level scientists a month or sometimes more to create. That's laborious, and that's not clinically useful time often in the sense that it takes too long, but it is important accounting of what we found. And then there's the targeted gene report, and that's the thing on the top. And the targeted gene report, the ambition is to report it out in 2 weeks, which we do at a fairly regular rate. That's good. That is clinically useful. And it would be better if it was 10 days. Of course, it would always be better if it's faster, but there's realities of turnaround time on the technology side of things. So here's the thing. A targeted gene report you can think of as essentially a fancy panel. Any new variant that comes along that's associated with some treatable element of cancer can be included electronically in the targeted gene report. The data are there. We just have to go look for it. So it's not a new assay. It's the same assay. Doesn't require any iteration in cycle times. We go looking for it straight away. So that it's an efficient way to incorporate new knowledge that emerges from the community into the reporting structure. Okay, so that's good. But we don't find many hits there. And many of the hits that we find are ones that the panels would find. By definition, it's a targeted gene report. We know about the targeted genes for the most part. So the hit rate there is maybe only 10 to 15%, depending on the series, depending on the patient, depending on the disease. From the big reports, what eventually emerges is an assessment by the oncologist that about 80% of patients have an actionable alteration, but only 30% are acted on. That's a big delta, and there's a few reasons for that. Somebody will ask me what those are at the end, I hope. Software is absolutely required, and to achieve this kind of turnaround and scale, there's a couple of software packages that have been produced, and we're trying to share these. They're sort of bespoke things and somewhat difficult to transfer to other jurisdictions, but we have a program in place now with Calgary, where the Marathon of Hope Cancer Center there is incorporating and testing this software package called PORI, Platform for Oncogenic Reporting. Our colleagues in the Atlantic provinces, Tony Ryman, is doing the same. So we're trying to spread this out across the network in some way. We're not going to go into it other than these colored boxes up in the top right show the IPR in action, and then the screenshot on the bottom is what a clinician can see when they log into the system and fire up the report. They can see all elements that we find in this particular way. The idea here is to have online access to the material that we generate so people can go back and reflect and see what we found, when we found it, how we found it, this sort of thing. And so that's available, and we're trying to disseminate it. And that's coming out of the Genome Center, particularly Steve Jones' work. So I want to have a couple of examples put before you here to illustrate how all this works in action. So, and the first example here is meant to illustrate that this is not just about targeted agents. We can use this kind of thing in chemotherapy, i.e. these broad spectrum cytotoxic agents that aren't focused on a particular mutation or an alteration. So in this particular example, the big red arrow is pointing to a gene called DPYD. And DPYD is a gene that we know about in the germline. So if you have a deficiency in DPYD in your normal DNA, you should not be given 5-FU. And the reason you should not be given 5-FU is because it will hurt you. DPYD is necessary to clear 5-FU. So if you're deficient in clearing 5-FU, if all your cells are deficient, there's cell toxicity there. That's bad. There was a highly publicized case maybe a year ago of a physician that passed away because nobody had looked at the normal DNA, and this individual had a DPYD deficiency. Okay, so what I'm showing there is this hash line through DPYD. That means there's a rearrangement of DPYD, and the blue color, which you probably can't see, indicates it's a predicted loss of function. And because we have the genome and the transcriptome, the structure of what we find is shown on the right. There's the DPYD locus with the intron-exon organization illustrated. And then there's this faint area bounded by 2 lines. That's an internal deletion. And this internal deletion takes out a chunk of DPYD and it produces a loss of function variant, or it's predicted to. Okay, so DPYD loss is associated with toxicity, 5-FU induced toxicity. This individual's normal DNA is fine, but their tumor is DPYD deficient. And so the hypothesis was that DPYD would act on the tumor and produce a cytotoxic effect there, but ought not to act on the patient, normal cells. And that's indeed what was found. And it was quite dramatic. So the patient was on an extremely expensive immune checkpoint inhibitor over a long period of time. The tumor blew right through that. And then this very inexpensive off-patent stuff was offered, and it resulted in a rather incredible involution of the tumor mass, which was on an unsightly thing on the neck. So it's not just about expensive. It's about understanding the profile and trying to align what you think you know about the biology related to the gene to a therapy that may be available. And this might actually be the best future for POG. Maybe we don't want to be focused on the really expensive things that are hard to get and offer patients. Maybe we want to focus on this kind of stuff. Don't know. Another example it's a different one. It's pediatric cancer now. And in this case, an immune checkpoint inhibitor was used. And we know that immune checkpoint inhibitors block the normal response of the body to its own immune system. So we want to protect ourselves from our immune system. It's a powerful thing. It can kill us. And so the body has in place these checkpoints, which tell the immune system to stay away. Don't attack me. And so tumors have hijacked this. They are us in some sense, and they cloak themselves with immune checkpoint molecules. And immune checkpoint inhibitors interfere with that signaling and expose the tumor to the action of the immune system. So here's a pediatric cancer patient. Now, this patient had some interesting features in their genome. They had 50 structural variants, so larger scale rearrangements, but 0 single nucleotide variants, none. What they did have was a heterozygous copy number loss and very low expression of a gene called SMARCB1. And this gene, aside from its admirable biochemical properties, has been associated with malignancies for a very long time. And so we have loss of that thing or partial loss of it. Right next door, we have very high expression of a gene called T or brachyury in mice. And we know that brachyury is normally expressed very early on in development, maybe even before the formation of the immune system. Is that right, Kathy? Yeah, Kathy said yes. So, and Matt's backing you up. So there we have high expression of something that we might predict to be an antigen in the case of, you know, an 8 or 10-year-old individual. And we have a goof up in how SMARCB1 functions. So those are the 2 key observations. The next key observation is shown that by that big bright red dot. So when we sequence the RNA, we infer that there are infiltrating CD8 cells, T cells in the vicinity of the tumor cells. And then in the blue box over there, we have high expression of checkpoint molecules. And so the logic here was we have a lot of T cells. They're apparently not acting on the tumor. We have a candidate antigen, P, and we have very high expression of immune checkpoint. So let's knock back the immune checkpoint with immune checkpoint inhibitors, expose the tumor cells to the action of the T cells, which are in the environment, and see what happens. And so nivolumab was given. This is the work of Rod Rosick and Rebecca Dale at BC Children's Hospital Research Institute. And the patient experienced a durable response to this. So these are anecdotes. You see the problem. The problem is cancer is an individual disease. Maybe, you know, there are some large number of cancers that we can treat in a uniform way and achieve good results. And we know what some of those are. That's true for standard of care. And then there's a whole host of cancers we don't really know what to do with. And maybe it's going to be individualized solutions for those. How do we recognize which is which? Important thing to think about. This extends beyond the anecdote. And so here is a class of rearrangements that were found across a variety of different cancers. The main gene is a gene called NRG1, and NRG1 in these cancers is fused to something else. Very often, it's fused to SDC4 or ATP1B1, what have you. And so we saw this initially in a lung cancer case. That's the second one down there. And when we looked at the fusion, we anticipated that the fusion between the 2 genes involved yielded an open reading frame. And the prediction was based on the RNA profile, which is shown in the histogram to the right. You see that red line? The red line is the expression of this case measured against the expression of NRG1 in all of lung cancer. So it's an outlier here. It's highly expressed. It's clearly a fusion, and it may have an open reading frame and retain function. So when you look at that and you look at the axis through which NRG1 acts and you look at the data, it looks like it's driving down through the MAPK signaling cascade. And it looks like, based on what we know about NRG1 biology, that it's acting through the ERB family of receptors, which sit at the cell surface. And so the decision was made to offer an inhibitor of the ERB receptors. And that, it's a dirty inhibitor. It inhibits bunches of them. And that thing was called afatinib. And so after much wrangling amongst the clinicians, the decision was made to try it. And there's an effect, and I'll show you a display to prove the point in a second. We then found an NRG1 fusion in a type of liver cancer, cholangiocarcinoma. The 2 had very, very high expression. Afatinib was recommended based on the commonality of the expression and mutational profiles to the lung cancer case. Showed good effect. And then it was found in KRAS wild type pancreas cancers. And not one, but a number of them. And so the NRG1 fusion class and its association with afatinib sensitivity was quite a finding, surprising to us. This is the lung case that I was talking to you about. There's a mass before treatment at baseline. Afatinib started July 16th, it says, and then a couple months later, there's fairly significant reduction in mass, and the patient felt better. We did not cure this patient, or really any of them, with some very rare exceptions, because these are very advanced in their disease. But we offer benefit. So here's a little picture of how the NRG1 fusions are thought to operate. At the front, they're signaling through ERB receptors to activate RAS MAPK. And it turns out that the KRAS wild-type pancreas cancers are only a sliver of the pancreas cancer pie, and that's what's shown under the pancreatic ductal adenocarcinoma case there. So you have a very small, 8 to 10% of PDACs being KRAS wild-type. And maybe that's what precision medicine means at its best. Maybe we're subsetting cancers based on their profiles into areas of biology that can be individually targeted. So maybe it's a little bit better than anecdotal, but maybe not a lot. Okay, so where are we today after this stroke of good fortune, if you like, 15 years ago? There's an accounting for things. Now, when I think about POG, it's become a platform, a way of doing things at BC Cancer, and various projects are bolting onto it now to take advantage of the report format, to take advantage of the community that's been built. And this, I think, is the important product, the community. And so in point number 2 there, it says 171 medical oncologists or fellows have enrolled patients into POG. And in 2024, there were 433 referrals from 102 practitioners. I think POG, and this I think is probably its most important contribution, has trained a cohort of clinicians to appreciate complex biological data, to evaluate it critically, to use what they can and learn from what they can't. It's not important that this be about genomes. It's important that we have a very sophisticated cohort of such individuals in our jurisdiction as the vanguards of cancer care. That's kind of incredible. And these same individuals would have the capacity to evaluate large-scale high-throughput proteomics data. They would have the capacity to think about all manner of these people have learned. This is transformative in my view. So we have a vision, and this is a heroic vision. It's direction setting, but probably unachievable at some level. But we imagine that by integrating with clinical data and linking through to clinical trials, we're going to be able to identify a cohort of very high-risk patients and do all of those. And I think the big costs here are not the sequencing. I think that's a distraction, and I think it's an argument that should not really dominate conversation. The sequencing costs are constantly in decline. Even a single high-throughput box could almost do a population, maybe 6,000 cases at the level of POG, 20,000 whole genomes kind of thing. So it's really not about the cost of doing the assays. It's the business of what it precipitates downstream. And when you put it into the cancer care continuum, you would be adding costs to an already struggling system. And so what are you not going to do in the system if you do this? This is an important consideration. All right. So we didn't really know how to carry this forward. We've reached a point where it's expected that we will do this. The clinicians have a data expectation. They want access to it. They expect us to be there to do this thing. But how do you carry this forward in some way? And so the carrying forward piece happened courtesy of the gentleman in the middle there. This is Dr. Victor Ling, who is the founding president and scientific director of the Terry Fox Research Institute. And Vic's retired now, but he's still active in the environment. Here he is standing beside Dr. Amelia Lim, whose laboratory is somewhere in this building. But of course, I can't come up and visit Kathy or Matt or even Amelia because I don't have the key card. So I don't know, where Amelia's lab is, but Amelia graduated from our lab many years ago. And it's gratifying after a stint in Cambridge for her to come back and receive the Victor Ling Terry Fox New Investigator Award. So that's Amelia and her labby there. But Vic started something called the Marathon of Hope Cancer Centers Network. And in this context, worked with the Harper administration, then worked with the Trudeau administration #1, then Trudeau administration #2, and then sort of in all of that, eventually managed to secure a commitment from the government of Canada. And so this was a commitment of $150 million to start the Marathon Hope Cancer Centers Network, to be matched 1-to-1 by partners across the country. So a $300 million program. So Vic got the money. And the idea that eventually emerged was, well, we can form this thing called a network, but it's meaningless unless it does something. So what should it do? And the decision was made that what it should do was focus on data sharing, and particularly genomic data sharing, because genomic data sharing was consumable by oncology communities. And that's what POG had given the Marathon of Hope, was a demonstration that, you know, this just wasn't necessarily a stamp collecting it could be interpretable in a clinically meaningful way. And so the project eventually launched, and relatively rapid time by government standards, perhaps. So in 2021, the contribution agreement to Health Canada was signed, and it's apparently a document that is fairly strict in its interpretation of how and when one spends money. So that contribution agreement launched the program. So what does the program do? There's our patient diagram there. It's got whole genome, FFPE, formalin-fixed, paraffin-embedded. So this is the type of material we normally see collected in clinics. It's got transcriptome, and this can be from formalin-fixed material or fresh frozen. And this is the tough bit, it's got clinical data. So the clinical data has to come from the healthcare organization. It has to be abstracted from what the organization collects, often on paper, and transposed and shared in the context of the network, which stretches across the country. To inform all of these processes and things that need to be done, working groups were formed. There are a large number of them. I won't read them all out, but just about every single thing that needed to be done involved the formation of a working group and consensus building across the country. This all took enormous effort and time, and bent a lot of the rules and practices in place of the various organizations. Because data sharing across constituencies, this kind of data sharing across constituencies, very, very hard, but not impossible, it turns out. And that's one of the things the Marathon of Hope Cancer Centers Network program has shown. The one point I will point out here is the formation of the patient working group. This is a highly engaged and very powerful group of individuals who are determined to see advances in cancer care, and are, I think, a huge resource for all of us that want to advance care to the point where we can offer better to our patient populations. So the patient working group needs to be called out and acknowledged for their absolutely incredible influence that they've had in the broader landscape. Marathon of Hope is rolling along. We're coming to the end of the first phase. March 31st, we have to hit 15,000 cases. And this is where we are were as of yesterday. So we'll probably get our 15,000 genomic data sets, but the clinical data sets are lagging, and this would be bad. We need to finish the job. And it's hard. It's the single hardest thing, not the genomics, not the informatics, but interfacing with the clinical systems and getting these data. We see 5 consortia. We see 54 member institutions. People continue to join. And to be a member institution, you just can't sign up. You got to be there to do stuff, give money. 141 cohorts. And along the way, we're funding clinician scientists and young investigators to think about precision medicine here in cancer, in Canada. We wrote a little paper describing the network. You can read about it if you want. And within this, there's some nice diagrams and a complicated diagram. This is the complicated diagram. And we're not going to go through it. It just sort of illustrates the data modalities that are collected. The only point I want to make is down at the right-hand corner there, where you see all the little Canadian flags. What you can't appreciate is that's superimposed on an invisible map of Canada. Those are the individual sites. And what was eventually decided as a compromise position is that we would have federated data sharing, so that each jurisdiction could decide what elements of clinical data it could legally share within the bounds of its, and that worked. That's the magic sauce, allowing a federated data sharing system. Okay, to test the data sharing system, TFRI launched a competition. And the competition was really to assess whether or not the thing they thought they had set up was actually usable in the broader context. And they wanted this within the network. 5 projects were funded. 1, 2, 3. Yep, 5 projects were funded. We applied because we wanted to help Terry Fox understand the data sharing system. And we thought we'd focus on glioblastoma. Why? Because we knew exactly what number there were in the country that had participated, and there were 120 cases. And we thought that was a small enough number that we could feasibly download the data, do something in very tight timeframes. This is basically a 6-month program. Okay, let's back up. Here's the critical thing. So if you look at a collection of POG cases, you can see in 570 patients, something like 7.4 million somatic substitutions, 750,000 small indels and a bunch of structural variants. The average number of somatic alterations in a cancer patient is 13,000. We'll be lucky using standard approaches to find a targetable alteration, even one in a patient. There's a huge mutational burden sitting there that is not being utilized or even thought about as a source for candidate alteration. So we have an idea, and the idea rests on the concept of synthetic lethality. Am I good until 2? When should I shut up?
1:50.
Okay, we might make it. I doubt it, but we might. So synthetic lethality. Who here has heard this concept? Okay, good. Some people have. For those of you who haven't, and especially for those of you who have, who can actually criticize this diagram, there's a car. The car can take either fork in the road and get to where it needs to go. It's got 2 pathways available to it. Well, if one of those has a great big boulder sitting in the middle of it, it can't transit that path. It has to take the other. Imagine that the boulder there is a mutation. So it's a mutation that exists. You observe it. All right, so there's a mutation there. The cell can't go there, but it can take the other. But what if you close that path and the cell can't transit either of those things? Can you kill it? So if these are viability pathways or pathways that arrest the propagation growth of the cell, can you kill it? I'm telling you, there's 13,000 of those in every single cancer patient-ish. So what about finding things that go there? How do we do that? And that's the concept that drove our application for the data sharing. So we have our contributing groups, Alberta, Atlantic, and BC a little bit, 120, I told you about that. I'm gonna talk to you a little bit about our synthetic lethal approach, but it starts with identifying mutations, obviously, so we need all of those. And then it uses a resource called DEP map, the dependency map. I'll describe that. We match the patient to these cell lines in DEP map, and we can predict synthetic lethal targets using a tool called Greta, which you can get off of GitHub right now if you want. And then we can align the outputs to approved drugs. That's the plan. Now, DEP map is a very useful entity. And depending on when you go look for an update, you might find more or less cell lines in there. They vary, but it's around 2,000, 1,600 to 2,000. And what they've done is they've sequenced the cell lines, transcriptomes and genomes. They have done some drug screens, done some proteomics, done some methylation, but importantly, they've done gene knockouts, 1-by-1 gene knockouts. So they've taken a cell line, they know what the mutational profile is, and then they can do some synthetic lethal screening, and they can look for second site hits, and they measure the viability of every single one of those constructs. And all those data are available for download. That's incredibly powerful, we think, for purposes of our synthetic lethal screen. So Greta is just an R package. Basically, it's a zillion t-tests, to think about it in another way. And so this allows you, Greta, allows you to specify the depth map cell lines you want to look like. So we're going to worry about GBM cell lines. So we want the GBM cell lines. We want to know all the loss of function alterations in the patient which we can then align to find depth map cell lines with the same mutation, and then we can query those cell lines viability data and look for second hits that impede viability. I have a cartoon. But before I get to the cartoon, this is the work of Yuka Takamon, who's done all this stuff, and we've applied it now in 4 different studies. We know it can give results when we dig very deeply into the nature of those results. Okay, there's our GBM patient, 1 of 120. We can find all its loss of function mutations. That's just the sequencing of the patient. Marathon of Hope does that. And we can take that 1 mutation at a time, and we can then go ahead and look for, in the first depth map cell line, we can look for a mutated gene. That's on the top axis. And then we can look for knockout genes that impede viability. And that's the red dot. So that's a combination of a mutation that we found in the patient that we also find in a GBM depth map line with a second site hit that impedes viability of that cell line. And what is that thing? Well, that thing is a candidate synthetic lethal hit. So then the question becomes what proportion of those actually can we act on? And we don't know until we've done the experiment. Okay, Marathon of Hope's coming to an end. I think the initial phase of the program will be judged more or less successful, depending on the clinical data acquisition. The next phase is coming. There's a video out there. It's accompanied the launch of something called Finish It, which is a national fundraising campaign. If you guys want to see an incredibly powerful depiction of AI makes me cry for lots of reasons, but mostly Terry's depiction, it's not AI, it's very clever standard photographic manipulation apparently. But there's an image of a runner who played Terry in this video, leading this cascade of individuals running forward. Go watch the video, you'll like it. It's all about prospective enrollment of patients and return of results. It's about POG. So it's really now trying to say, can we do POG nationally? And how can we here in BC help? Because if we can get the OICR, the Ontario guys cooking, if we can get Montreal cooking, you know, the reach will extend beyond what we can do here. And we're enthusiastic about that. The ambition, obviously, is to deliver precision cancer omic medicine to Canadians. That's what we want to do. And we've got 4 years to make the research that we think will be required to get jurisdictions interested. This has to be fairly deliberate and well thought out. I have no idea how we're going to do it. But anyway, it's a good challenge. So we have lots of members of the Prospective Enrollment Working Group. Janessa Laskin is the chair. So there's POG again and some other members. So we need prospective enrollment. This isn't somebody's collection of tumors sitting in a freezer. It's a new patient, walks into the clinic, has a need, and they're enrolled. The expectation is we're going to do stuff and quickly. And then we're going to have to think about reports. And so there's the PORI system that helps generate reports. Maybe that's going to be useful. There are other systems out there, including Trevor Pugh at Princess Margaret Hospital has generated something called JERBA. Okay, so in closing, I think the Marathon of Hope is our opportunity to drive POG forward in some convincing way. I don't think this is the end of the idea that advanced measurements would come forward and influence treatment planning for patients. It's the beginning. More and better will follow, but it's a start. And data sharing is fundamentally important, although it's tough. Thank you to the patients. None of this would happen without patients. And the strength of the patient working groups is truly something to behold. We're all suspect, us researchers, because it's self-serving for us to go to Ottawa and talk about these kinds of things. But the patients have a different perspective, and they have power. I'll point out Yuka. There's Yuka, who did all of this formulation of Greta, and Richard, who's helping Yuka. And there's Alex. And Alex is a master's student who is learning about Greta. And unfortunately, Karina's not there. Karina is also learning about Greta and working on GBM. Thanks to all of our funders. UBC is a partner in all of this. It's fantastic. BC Cancer Foundation funds POG alongside Genome British Columbia, and the Terry Fox Research Institute is driving us all forward. And with that, I will close. Thanks very much, folks.
Thank you very much. Were there any questions?
Thank you for the great talk and bring us a great vision of the new future kind of treatment for cancer. I have two questions. One is does the POP program also apply to some aggressive cancer like pancreatic cancer or highly mutation cancer? Because the turnover, does it kind of okay for the patient to get treatment? Because given the genome sequencing might take some while. And the second question is, does it also apply to metastatic cancer? Like, because for the biopsy, do you take from different location to do the sequencing? Thank you.
Yeah. So does it apply to pancreas cancer? Absolutely, it does. And so there's a program. So Dan Raynoff is a leader of POG, and he also leads a pancreas research effort, and he treats pancreas cancer patients in the clinic. And so we sequence pancreas cancer patients in POG. So that yes is the answer to that. And then multiple biopsies. So this is a really critical question, actually, it turns out. So what influences the selection of biopsy sites? The number one consideration is accessibility and safety. And so if you can't stick a needle in it safely, it's not going to get done. And even if you can stick a needle in it successfully, sometimes it isn't done because of financial constraints. So none of this is done with a budget that would accommodate those kinds of questions. That's something that should be addressed. I don't think it'll be addressed in the next phase of the Marathon of Hope. It could be addressed in the context of POG. We've seen lots of cases where we have discordant responses to treatment in patients. One metastatic node will respond differently than another. These discordant responses are the proof that what you say we should be doing, but not yet.
2,300 POG participants, how many were blood tests?
Historically, not a lot, actually. So I think there's some AML in there, but not a ton. Not a ton. It's all driven by clinician interest. So POG is an open program in the sense that people that have an interest, 103 people. So you can enroll onto POG, you can bring new programs into POG. Some people have argued that some of the outputs aren't really relevant to their kind of cancer. It depends a lot on what the clinician wants to get out of it. Some clinicians participate and enroll cohorts because they want to learn more about the patients they're treating. Some clinicians are simply asking the question, what can I give my patient? But in some cases, there's not much that we can do. And a good example of that is GBM. You got the blood-brain barrier. You've had really no meaningful innovation in therapies since temozolomide, which is ancient and contributes very marginally to lifespan extension. And I think we have to do special things for those kinds of cohorts to understand what the clinical body wants to learn. What I'm hoping to enrich collaborations with GBM and perhaps others is it will be able to leverage some of these other mutations to expand the number of therapies that we might consider for a patient. And maybe there'll be stuff in there that does penetrate the blood-brain barrier, or maybe some new innovation will emerge in the space that we'll be able to capture and tag with a genomic signature that we can recognize. So will we accept blood cancers? Yes. Do we have many of them? No, but that's clinician preference, I think, more than any other thing. Yeah.
Yeah, that's really good, interesting overview of the whole program. Who pays for this? Does MSP, are these billable items?
No.
It's all through grants or the program?
Yeah, yeah. So for the longest time, it was BC Cancer Foundation, and then Genome BC chipped in some money, and now the Marathon of Hope Cancer Centers Network is funding a very significant amount.
So what's the real cost? Not the cost to be put in grants, the actual cost of the sequencing and whatever other it is per patient or per analysis.
Yeah.
Just so we have a ballpark.
Yep.
figure in our heads.
Yep, so the cost that.
And then not the stuff in the grants, right? You know, the research that goes with just.
Oh,
the
no,
actual-
we understand the real
Yeah.
cost.
Yeah.
So when the program started, Terry Fox said, we will give you $6,000 for sequencing, and you will provide $6,000 for whatever it is that you need to get the clinical data. So it was a $12,000 price tag for patient. We think, that was 5 years ago when the program initiated. Today, we know that the real sequencing cost is around about 3. The clinical data costs aren't driven by any element of this other than how cancers are managed in the local environment.
Sure.
So we have no influence on that. But I will tell you that those costs have not decreased, and the costs of sequencing and processing have, to the point where the dominant cost is actually human beings running around collecting clinical data, for example.
So for the cancer board, do those clinicians, do they bill MSD for their time?
Oh, I'm not one of those people, so I would imagine that they're paid, but I think part of their contract is for research time, and I don't know how they divide it or think about that. Most likely, but I don't know. I'm just happy they're there.
Yeah.
Looks like we are past the hour, so Marco, thank you very much.
Is this meeting still open or is it closed?
I think it's done, Cedric.
Okay, thanks a lot. I did have a question, but I can talk about it all the time. Thank you.
For my back. Yeah, please do. Do you want?
Find ways to be out. Right. Okay, thank you, Matt.